For centuries, biology was all about what you could see through a microscope. Petri dishes, lab coats, the whole deal. But that's all changing, and fast. Today, biology has become a science of code, of algorithms, and just massive, massive data sets. So today, we're going to dive into how scientists are becoming digital detectives, mining this biological code for the next generation of cures. This one sentence really just says it all. It perfectly captures this gigantic shift in how we approach science. The study of life isn't just about looking at living things anymore. It's about trying to decode the absolutely insane amount of information they're constantly generating. And when I say the amount of information is insane, I'm not kidding. The first thing we really have to wrap our heads around is the scale of the data we're talking about. It's not a stream, it's not a river, it is a full-blown tsunami. Okay, so most of us have heard of Moore's Law, right? The idea that computer power basically doubles every 2 years. Well, check this out. Genomics is on an even faster track. The amount of public sequence data we have doubles every 16 months. So the data is actually growing faster than the technology we built to analyze it. It's just wild. To put a number on that, just one single database, the European Molecular Biology Laboratory, holds over 1.7 trillion base pairs of sequence data. That's a number so big our brains can't really even process it. We're talking about a library of life's code that is just incomprehensibly huge. So this brings us to the big central problem, right? When you have a haystack of data that big, how on earth do you find the one tiny needle, that one genetic mutation, that one protein interaction that could actually lead to a breakthrough? And here's what makes this so incredibly tricky. Unlike a lot of other data problems, biological data is super wide. We're talking thousands of features, like genes and proteins for every single sample. But it's also very shallow. You might only have data from a few hundred patients. So you end up with more questions than answers, more variables than examples, which makes finding the important signals almost impossible. And that is where biological data mining comes in. Think of it like a set of superpowered, high-tech metal detectors. These are the computational tools that are specifically designed to sift through all that noise and find those crucial digital needles. So what's actually in this data miner's toolkit? What are these algorithms doing? Let's take a look at just a few of the techniques that are turning all of that chaos into real clarity. First up is a technique called classification. The easiest way to think about this is like a sophisticated sorting hat from Harry Potter. Its entire job is to learn the difference between, say, a healthy cell and a cancerous one, so it can then correctly label any new cell it sees. Now, one of the most clever ways to do this is with something called boosting. It's pretty genius, actually. The computer starts with a really simple, weak model. Then it looks at what it got wrong. For the next round, it gives more weight to those mistakes, basically telling the next model, hey, focus on this hard stuff. It repeats this over and over, and by combining all those simple models, it builds one super accurate, strong classifier. It literally learns from its mistakes to get smarter. All right, next up we have network analysis. Now, this isn't about sorting things into buckets. This is all about relationships. This technique builds what's basically a social network for genes and proteins, so we can map out who's talking to whom, who's influencing whom, and which ones work together as a team. And what's really fascinating here is this idea of graphlets. Think of them like Lego bricks. Scientists found that these gigantic, complex biological networks are all built from a small set of these recurring connection patterns. So it's like realizing all these massive structures are made from just a few simple Lego pieces. If you can understand the pieces, you can start to understand the whole thing. This third tool is one of my personal favorites, text mining. Because see, the data isn't always inside a cell. Sometimes it's hidden in the millions of scientific papers that have already been published over decades. Text mining algorithms are designed to read this entire library and find connections that no single human could ever find. And this is how it pulls it off. The algorithm basically turns every scientific paper into a kind of numerical signature. It converts messy, unstructured text into clean, structured data a computer can understand. This lets a researcher ask a question like, show me every paper that mentions this specific gene and this specific disease. And the computer can find connections that a human, even reading for a lifetime, would totally miss. So we've got these incredible tools, but what's the payoff, right? Why does any of this matter? Let's get into the real-world impact of all this. Okay, let's start with a question that affects millions and millions of lives. How can we use all this data to look at a tumor and not just say, yep, that's cancer, but to determine its exact subtype, to predict how aggressive it's going to be, and to pick the absolute best treatment for that specific person? This is where data mining is already making a huge difference. We're talking about algorithms being used right now to classify tissues as healthy or diseased, to find tiny molecular biomarkers that could help us detect cancer way earlier, and to untangle the messy web of gene interactions that cause common diseases. They're even starting to predict which drugs will work for which patients, which is the foundation of true personalized medicine. But of course, with stakes this high, we're talking about people's health. You have to ask the most important question of all. We've built this cool predictive model, but is it any good? Is it reliable? How do we even know we can trust it? This whole process is called validation. One of the gold standards for checking accuracy is something called the AUC score. It's really simple. A perfect test gets a score of 1.0. A test that's basically useless, no better than flipping a coin, gets a 0.5. For a model to be considered good and potentially useful in a clinic, you really want to see a score of 0.8 or higher. It's a simple, powerful scorecard to know if a model's actually working or not. So where is all this heading? What does this mean for the future of science and medicine? I think this is the single most important thing to take away from all this. These tools aren't just for organizing data anymore. They are becoming an engine for discovery itself. By finding these weird, unexpected patterns, data mining is actively generating new ideas, new hypotheses for scientists in the lab to go and test. The data itself is starting to point us in directions we never would have thought to look. And that leaves us with one last, pretty profound question to think about. With trillions of data points already collected and sitting on servers around the world, is it possible? Could the clues to curing a major disease already be out there, just waiting for the right algorithm to connect the dots? It's a huge thought to end on. Thanks for joining me.